Cool. Hi, everyone. I have the pleasure of introducing John Doench. Dr. Doench is an institute scientist as well as the director of research and development at the Genetic Perturbation Platform at the Broad Institute of MIT and Harvard. Prior to this, he graduated from Hamilton College, received his PhD from the biology department at MIT, and completed his postdoc at Harvard Medical School. In 2013, Dr. Doench received the Broad Institute Excellence Award in Collaboration and was named Merkin Institute Fellow. And in 2018, he received the Broad Institute Award. Dr. Doench and his team worked together toward the development of functional genetic techniques that utilize RNAi and CRISPR technology. They have developed cutting-edge bioinformatics tools as well as libraries for CRISPR screens that have been used in numerous publications. Today, he will be presenting on his functional genomics toolbox. So thank you, Dr. Doench, for joining us. And with that, I'll hand it to you.
Thank you so much, Sadie, and really thank you so much for the invitation. I couldn't travel to Memphis this time, although I visited Memphis right before the pandemic. It's a really lovely place, and I hope to maybe I'll be able to visit before some of you graduate. But don't wait on that. Get your PhDs as fast as you can. Okay, so I'm going to talk about the functional genomics toolbox. And I can lecture for the whole time, but I think it's a close enough audience that if you have questions, certainly feel free to just unmute and shout them out, and then we can talk about them. This doesn't need to be a monologue, but of course we'll have time for questions at the end as well. Okay, so our genome is a bit of a dumpster fire of information. So by way of analogy, let's say you went to IKEA and you bought 20,000 things, those 20,000 things being an analogy for the 20,000 or so protein coding genes in the human genome. But then you got home and you mixed all the parts together on your living room floor, and then you burnt the instructions. So now you've got all the pieces to make your Klondorf and your flock dip and a lot of Allen wrenches, but you don't know how to put them together. And our genome is not all that dissimilar from the standpoint of it's the product of 3.5 billion years of short-term thinking. It's a genome that has evolved over time. There's a lot of redundancy. There's a lot of stuff that we don't know how to make sense of, and trying to understand how small changes in our genome, an A instead of a G at this position gives rise to a 2% increased lifetime risk of diabetes is not a trivial thing. And indeed, after the Human Genome Project was completed, one of the leaders of the effort was asked to summarize it in 7 words. And he said, genome, bought the book, hard to read. Or maybe put another way, you have this for 3 billion bases, but you can't just call up a helpful Swedish person to figure out how to make sense of it. So the question becomes not just what are the genes, but what do all these genes do? And how does gene dysfunction lead to disease? And that was sort of a fundamental question in biology now that we had the sequence of the human genome. And this is right around when I was starting my PhD career. And so how do you determine function? Well, one way to do it is to do a controlled experiment where you change or perturb one thing at a time. So let's say that something on your desk is beeping, and you don't know what thing is beeping, but you want the beeping to stop. So you just, crawl under your desk and you start to unplug one thing at a time until the beeping stops. And you say, okay, well, this must be the thing that was doing the beeping. Or you want to figure out, was it the quarterback or the coach that was so important? Well, you get rid of one, keep the other, and then you can get a pretty clear answer of which was more important to your football team's success. But how do you do this with genes? How do you manipulate genetic information? How do you break something or perturb something one thing at a time? Well, for right when the human genome sequence was published, really the only technology that we had in our toolbox to do this in mammalian cells was RNA interference, or RNAi technology. And this works by killing the messenger. The basic idea with RNAi is that you can put a small RNA into a cell. This small RNA directs a pair of molecular scissors known as the RISC complex, to cut messenger RNAs. So you're killing the messenger, and you can understand, okay, well, if I silence this gene or that gene or whatever gene, what phenotype does my cell have? And so this is what we did for quite some time to understand in an unbiased way, what phenotype do I get when I silence each gene one at a time. Challenge, though. It turned out RNAi was not that great a tool for the job. And that was summarized by this editorial written by Bill Kalin in Science magazine that appeared in just over a decade ago, entitled The Use and Abuse of RNAi to Study Mammalian Gene Function. And he begins his editorial by noting that the honeymoon is now over. Now, whether you're married to a human or a technology, that's not a great sentence to hear. And he noted that, even though some new discoveries had been made, the yield has fallen short of expectations. Drug targets identified by this technology in academic labs don't repeat when pharma picks them up. And there's a lot of pitfalls to the technology, but probably the most important is off-target effects. And that is illustrated by some data on the left. So these are data that we collected at the Broad. Each dot represents a different small RNA put into a cell to silence one gene of interest. And on the x-axis, we're plotting how much of the gene expression changes. So this was done with, it wasn't done with RNA-seq, but a similar technology to RNA-seq, where we're measuring lots and lots of mRNAs in the cell. And on the x-axis, we're quantitating the on-target magnitude. How much of what happens to the cell is explained by an on-target effect of the small RNA targeting the gene we hope to. And the y-axis is how much other unexplained effects happen in the cell, which we're calling off-target effects. And you can see that the problem isn't lack of on-target magnitude. Almost every dot is to the right of 0. The problem is the majority of the dots are above the diagonal, indicating that more of what these small RNAs that we're putting into cells to trigger RNAi, more of what they're doing is off target than on target. So they're not faithfully telling us what the consequence of silencing that gene really is. Fortunately, our time in the wilderness as a field of functional genomics was very, very brief because literally 3 weeks later, also in Science magazine, Jennifer Doudna and Emmanuel Charpentier published this paper on a programmable dual RNA-guided DNA endonuclease and adaptive bacterial immunity. And this is the paper that really described in clear detail how CRISPR technology could be made to work to target genes of interest. So from my perspective, this was great. I barely needed to update my slides. I just needed to move my scissors from cutting mRNA in the cytoplasm to cutting DNA, change it from an siRNA to an sgRNA, and we were good to go. But now we had another technology that we could use to manipulate genetic information and see what happens. And indeed, when we do the same experiment as we did before with RNAi, but now on the right, each red dot represents a guide RNA intended to cut a particular gene. We see on-target magnitude actually fairly similar to RNAi, but again, most importantly, almost all the dots are below that diagonal, indicating that the vast majority of what these things are doing to the cell is due to what we want them to do, targeting the intended gene rather than having some sort of off-target effect. Okay, so now we have a technology, CRISPR technology, and it really allows us to answer, I'd say, what are 3 of the most high-level, broad questions in functional genomics. And the first of which is, well, what are all the genes involved in my phenotype of interest? And to answer these questions, a really scalable approach that we love to use is pooled screening. Pooled screening starts with the synthesis of oligonucleotides that you then clone. So each oligonucleotide encodes a different guide RNA targeting at a different gene in the genome. So you can make these large-scale libraries, put them into lentivirus, then put them into cells, and then you subject your cells to either positive or negative selective pressure. And I'll give an example of that in a minute. And at the end of the day or the week or the month, you isolate the genomic DNA back out of those cells. You do PCR to figure out how did the abundance of my guide RNAs change over time, or the selective pressure. You Illumina sequence it, and ultimately you get your hit list. I just want to point out that there are three technologies that this stands on, and each of these technologies is a field of development in and of itself. The ability to write DNA in massively parallel format. Obviously, that is critical to a lot of biology now, but it hasn't always been that way. Likewise, the ability to deliver nucleic acids stably to cells. That's lentiviral technology, which was developed largely in the 1990s. And likewise, Illumina sequencing or next generation. It's been called next generation sequencing for like 20 years now. But the ability to move beyond Sanger sequencing and read lots and lots of DNA inexpensively is also really critical for this process. So I think this is just a good example of how technology development continues to enable new things, and any new technology tends to stand on the shoulders of some prior technology developments. Our model in the GPP at Broad is a very collaborative one, where there are some aspects to this process that scale really well. And so those are the ones that are highlighted in blue, where there are others that don't scale particularly well and don't need to scale particularly well. The exact genetic screen that someone wants to do. That is something where the collaborators that we work with, and we work with many, they can do those parts of doing lentiviral transduction, applying a selective pressure, and isolating genomic DNA. That doesn't require any specialized robotics or equipment setup or anything else. So the majority of the screens that we do, GPP, we do some of the parts, and our collaborator does some of the other parts, and these handoffs are actually pretty seamless. In the year 2023 alone, we made lots and lots of custom libraries, lots of lentivirus, ran a lot of PCR, and collaborated with over 100 PIs in the Boston environment and beyond. So I want to give an example of how these screens can work. So cancer immunotherapy is certainly one of the most exciting developments in the world of cancer biology over the last decade or so. And it arises from the basic idea that a T cell normally is recognizing tumor cells as foreign. And so our immune system is routinely clearing out incipient tumors in our body all the time, and that's great. However, the tumor cell, unsurprisingly, learns some tricks, and it figures out how to put the brakes on this T cell mediated killing. And one of the most common ways it does that is by expressing a gene called PDL1, which binds to the PD-1 receptor on the T cell and prevents the T cell from recognizing and clearing out the tumor cell. And so there's a number of therapies out there, the most probably famous being Keytruda, that blocks the interaction between the T cell, or blocks this repressive interaction between the T cell and the tumor cell, and now reallows the T cell to recognize and kill tumor cells. And these have been, it's worth pointing out, for historical purposes, this paper, The Hallmarks of Cancer, was published right as I was starting to get my PhD by Douglas Hanahan and Robert Weinberg. Everyone in this is a review that has been cited tens of thousands of times. Like everyone read this, and it was like, okay, here's our state of cancer. And you'll notice that avoiding the immune response was not on really anyone's radar in the year 2000. But it is now, of course, very much on people's radars as one of the most important paths for treating tumors. So this graph, this Kaplan-Meier curve, shows the promise and the challenge of immunotherapy. So this is this is for patients who had metastatic melanoma, stage 4 melanoma. And what is shown here is one of the first clinical trials of Keytruda. You can see standard chemotherapy, 50% of patients progress within just over 2 months, and essentially every single patient has progressed after a year. Whereas with Keytruda, you see that the line flattens out somewhere higher than 0. So this is amazing. So that's the promise of cancer immunotherapy, that in some patients, it really does seem to produce what we could call a cure. But of course, the challenge is that that line levels off at 20%, not at 100%. And the situation is much more dire in many other tumor types. Melanoma is actually one of the more responsive cell types to immunotherapy, but there are many, especially solid tumors, that just don't respond to immunotherapy well at all. So we wanted to ask the question, could we find genes that would enhance immunotherapy? So what did we do? We started with tumor cells and we introduced a library of guide RNAs into those cells. So it's just schematized here. Each cell is a different color because it has a different gene knocked out. So every gene is mutant in, or every cell is mutant in a different gene. We then take that pool of mutants and we introduce them into mice. We then either treat the mice with immunotherapy or as a control, we don't treat them with immunotherapy. Then at the end of a couple days, we remove the tumor cells back out of the mouse, and we then sequence the different guide RNAs that are present in those populations. And what we're looking for are examples like the red gene or the red cells, where those cells were fine in the mice that didn't receive immunotherapy, but then when you added immunotherapy, those cells were depleted from the population. And it turned out that there were lots of these so-called red genes in this screen. So what's shown here is a result from the first paper describing this, where here are all the genes, or a list of the top genes that led to this phenotype, where they were more depleted in the tumor cells when we had treated them with immunotherapy. And I highlight the gene PTPN2 because that's one that has subsequently undergone more study. It's a phosphatase, so it was potentially, and I should point out that here are the citations for the papers shown here. But PTPN2 was particularly exciting because it was a phosphatase. PTPN family members had been worked on already, and this pretty quickly led to a collaboration between the Broad and the pharmaceutical company AbbVie and another partner, Calico, to develop PTPN2 inhibitors and see how well they worked. And indeed, a small molecule inhibiting PTPN2 and PTPN1, a similar gene. This was published in October of 2023, showing the development of the small molecule. And there is a clinical trial enrolling patients right now to see if there will be any synergy between inhibition of PTPN2 and immunotherapy approaches. So this is all very exciting. Now, not every screen leads to a clinical trial being launched within about 4 years. But I think it does illustrate quite nicely what when everything falls into place, and you ask a good question and you have good reagents and you have good model systems, that you can really make progress quite quickly in a way that just kind of really wouldn't be possible in any other way. So I've talked about CRISPR or Cas9 to its ability to introduce double-strand DNA breaks and to cut DNA and to make knockouts. That's what Cas9 is doing natively in its job as a bacterial immune system. But you can actually tether lots of different domains to a Cas protein to make all sorts of manipulations to the genome, such as you can turn Cas9 into a transcriptional activator by tethering to it some transcriptional activation domains. Likewise, you can turn it into a transcriptional repressor by adding some repressive domains to it and turning it into a RNA-guided DNA binding protein. We have, at the Broad, we've made lots of libraries to enable genome-wide interrogation via not just CRISPR cutting, but some of these other modalities as well. When we first started doing this, we realized we were probably going to make a lot of libraries. So we started naming our human ones after wines, our mouse libraries after cheeses. We always empirically test that the wine and the cheese pair well together. And if they don't, we pop open another bottle of wine and we keep testing until we get it right. We're very committed to high quality wines here. Okay, so that's an example of how to start with a phenotype of interest and ask of all the genes in the genome, which ones are involved in my phenotype. But of course, a lot of times we already have a reason to be interested in a gene, a particular gene, and we want to understand how does function arise from my gene of interest. And just knocking the gene out is not that's a pretty blunt tool to understand gene function. So to get at gene function in a more defined way, we can actually use other types of CRISPR technology to do that. So what's schematized here is a Cas protein bound with a guide RNA shown in purple, bound to the DNA strand showed open up in the light and the blue, the light and the dark blue. Uh, and a couple of years ago, David Liu's group at the Broad developed what was called base editing technology. And here the idea is you're not using Cas9 to introduce a double-strand DNA break into your DNA, but rather you're positioning an apobec domain over the exposed single strand of DNA. And what apobecs do is they convert cytosines to thymidines. So this is a way of changing Cs to Ts in the genome in a very focused way. He then, the next year, developed a similar idea, but now instead of changing Cs to Ts using a different domain, one can convert As to Gs. And then a final tool in the toolbox that we like to use is here I've noted that for Cas9, guide RNAs need to be positioned next to what's called a protospacer adjacent motif or a PAM, which means you have to land a guide next to an NGG. But folks have evolved versions of Cas protein that are actually more flexible, where they only need to be next to an NG. And so what this practically means is that there are more guides that can work for a target site of interest because the PAM motif is fourfold less restrictive. You can have more guides targeting a gene of interest. So how does this work in practice? So let me give you an example. So we wanted to use base editor technology to understand how proteins interact with a particular small molecule. In this first proof of concept experiment, we wanted to study the gene BCL2, which is an anti-apoptotic protein, and a clinically approved small molecule inhibitor of it, venetoclax. So BCL2 is an anti-apoptotic protein. It tends to get upregulated in a variety of cancers. Obviously, many cancers are constantly receiving signals to commit apoptosis, but they evolve ways of overexpressing these anti-apoptotic proteins, so they don't. So venetoclax was developed as a way to basically turn apoptosis back on, but unsurprisingly, resistance evolves. So you treat a patient with venetoclax, you see a good response, but then resistance mutations arise. And that's what's schematized on the right is sequencing of patients who relapsed on venetoclax. And you can see here, so what's highlighted in the crystal structure are mutations that were found in these patients. And unsurprisingly, the majority of these mutations are really right in the binding pocket for venetoclax. So these are mutations that inactivate the ability of venetoclax to bind to BCL2. So we asked the question, well, could we have found this in advance? Rather than needing to discover patients who had relapsed, could we use base editing as a way to understand small molecule protein interaction? So to do that, we took a AML cell line, MOLM13 cells. We made a library of guide RNAs targeting BCL2, so every possible spot that we could land a guide, we put into the library. We then did a screen with either a C to T base editor or an A to G base editor, where we treat the cells with venetoclax or we don't. And we look for what are mutations that enrich when we treat with drug, indicating that these cells now have resistance to venetoclax. And we can plot the data across the length of the protein. So on the x-axis here is each amino acid residue that is targeted by a guide RNA. Each dot is a guide. And on the y-axis is the z-score from the screen. And then when we map those onto the crystal structure, we see that the majority of the high-scoring hits are also, as we saw in the patient data, are also right in the binding pocket for where venetoclax binds to BCL2. Here's just one example. So aspartic acid 103, a base editor mutates it to a glycine, and in doing so, instead of the red side chain forming a nice hydrogen bond with the blue small molecule, now that hydrogen bond is lost, and the small molecule doesn't bind nearly as effectively. So if we had no idea what the target of venetoclax was. We could have found it with a screen like this. We had no idea where in BCL2 this small molecule was binding. A base editing screen would have been able to tell us this. Let me give you another example. So P53 is the most studied tumor suppressor gene in cancer. It's the most frequently inactivated gene in cancers. So we created a library, a base editing library, Tylene P53, and then we have a no treatment arm as a control. We could also add Nutlin. So Nutlin is an MDM2 inhibitor, which leads to the stabilization of P53. And so in a Nutlin treatment arm, loss of function mutations will enrich. However, if you treat the cells with etoposide, then you actually need P53 for the cells to initiate repair from the DNA damage caused by etoposide. And so now loss of function mutations deplete. So we're able to look in both a positive and a negative selection direction. And so here are the results of the screen. Again, each dot represents a guide RNA. And one thing that's immediately obvious is, well, two things are immediately obvious. First, Nutlin and etoposide have different effects. You know, the data basically mirror each other. And the majority of the action is in the DNA binding domain. Most of the mutations that affected the fitness of the cells following either etoposide or Nutlin treatment are in mutations in the DNA binding domain of P53. And when you look at what actually happens in patients, this is also what you see. The hotspot mutations in patients with a variety of cancers are also in the DNA binding domain. So again, if P53 is a very well-studied gene, but if we were looking at a gene that we didn't know much about, you can certainly use these techniques to understand, well, what are the most important regions? What are the most important domains of a particular protein? Indeed, if you look at just the different domains that are annotated via of P53, you see that if there were the guide RNAs that targeted interdomain or regions that have no domain, none of them scored as hits, whereas the DNA binding domain, a high fraction of the guide RNAs scored as hits. So you could use this in an unbiased way to map a protein for which not much is known. These base editing screens also allow you to see some very interesting biology that would be hard to uncover in any other way. So if you squint at this graph, you can see that, well, the red and the blue tend to be mirror images of each other. When looked at broadly, at the very end terminus, the red and the blue dots actually march together. So Nutlin and the mutants created now actually do the same thing in the presence of Nutlin or etoposide. And this is a little more obvious when you plot the same data in a slightly different way. So now I'm just plotting the z-score achieved in the Nutlin arm for each guide. Each dot is a guide on the x-axis and etoposide on the y-axis. And we see that they're generally negatively correlated, but then there are some that fall off the diagonal. And we were interested in, well, what are these things doing that are falling off the diagonal? And so here's the results of two of those different guide RNAs. Two of them, whether you're using an A base editor or a C base editor, what they are doing is they're mutating the start codon of P53. In the case of an A base editor, they're changing the start methionine to a threonine. In the case of a C base editor, they're changing it to an isoleucine. And we validated that the results hold. Indeed, when you introduce these mutations, now either of these alleles enrich. So this was kind of surprising, but then we did some reading about P53. Again, it's a very well-studied gene. And it turns out that there is, in addition to the most normally used methionine at position 1, there's another methionine at amino acid position 40 for the full-length gene. And indeed, there are splice mutants of P53 that lead to translation not including the conventional start codon, but instead starting at amino acid 40. And what this does is it leaves off part of the first transactivation domain. So we're now creating a variant of P53 that now has different properties. And indeed, we also see this on Western blot. If we add in the guide RNA and then select with Nutlin, we see that an increase in this shorter isoform of P53. So this is an example of how we were able to study an endogenous element of translational bypass that would have been very difficult to uncover in any other way. So with these base editing screens, what's great is you can mutate endogenous elements. You can uncover drug resistance mutations. I gave an example of BCL2, but we've done this with a number of other small molecule protein interactions, including MCL1, BCLXL, and PARP. You can look at loss of function mutations and endogenous regulatory elements. We did this with P53, as I showed you. You've also done it with BRCA1 and a number of other genes. And I think the two best use cases of this technology is first, if you have a less characterized small molecule, which is very common in drug development, you're trying to target a certain gene and you have a small molecule and you think you know what the target of it is, nailing down what the actual target is actually really laborious, and sometimes you never really know. But I would argue that if you do a base editing screen and you can generate a point mutation that now shows resistance to your small molecule, that's really good evidence that you have in fact identified the correct target. I think another way you can use these screens is if you have an uncharacterized gene, and it doesn't even need to be protein coding gene. You could be looking at regulatory. You could be mapping enhancers. You could be mapping untranslated regions. I think base editing screens are a great way to just to mutate in a focused way a DNA region of interest and understand then how the cell responds to it. And in fact, there have been a number of base editing screens published just in the last couple of years that I think show really good different use cases of how one can use these very focused changes in the genome to get some really high resolution understanding across a variety of biological problems. Okay, so that's looking at the amino acid level or the nucleotide level for function. But genes don't operate in a vacuum. Genes operate by interacting with each other. So now let's take a giant step back and look at technologies that allow us to ask questions of, well, how do genes interact with each other? So genetic interactions. So one of the types of genetic interactions that is very interesting and impactful is the idea of synthetic lethality. So this is the idea that if you have two genes, gene A and gene B, if you mutate one of them, the cell is fine. If you mutate the other, the cell is fine. But if both of them are lost, then that gives rise to a lethal phenotype. And this has been particularly important in thinking about how to target cancer cells, because if we could find some gene A's that if the cancer cell is already mutant in gene B, it's already lost a tumor suppressor, could we find a gene A that would particularly inhibit the viability of the cancer cell, but not the normal cell, because by definition, the normal cell hasn't lost the tumor suppressor gene, and the normal cells would be spared. And indeed, remember, the vast majority of mutations in cancer patients are loss of function mutations of tumor suppressors, not gain of function mutations in oncogenes. And it's really hard to think about how you would develop a small molecule that targets something that's not there, right? You can't target a tumor suppressor, it's not there anymore. So this approach for thinking about how to target cancer, this was first really formally described by Lee Hartwell and Steve Friend and colleagues in 1997. The first poster child for this was the recognition that inhibition of PARP would selectively kill cells that were already mutant in the BRCA1 or BRCA2 genes. That was shown in 2005. And the first clinical therapy, the first clinical approval for this general approach was a PARP inhibitor that was first approved in 2014, and there are now a number of PARP inhibitors on the market. So to study combinatorial interactions, one can use Cas9, and we've done Cas9-based combinatorial screens quite a bit. But actually, our preferred tool for that is a different Cas enzyme known as Cas12a. Remember, CRISPR systems are found across all sorts of microorganisms. Cas9 is not nearly the only tool that evolution has come up with, and Cas12a is just a different CRISPR system from a different microorganism, and it has some fundamentally different properties. So first, key to that is the Cas12a system. All the different guide RNAs that it uses are expressed from a single transcript. And the Cas12a protein has two enzymatic functions. The first is an RNA-guided endonuclease domain, where it recognizes a constant stem loop sequence, binds to that RNA, and liberates each individual guide cassette. As schematized here, we now have 3 different sequences, the blue, the red, and the green, each of which has a different DNA sequence, whereas the stem loop in black is a constant sequence. But then, like Cas9, Cas12a can use that guide to find complementary DNA, open it up, and introduce a double-strand DNA break to inactivate genes. We particularly like to use Cas12a for doing these combinatorial screens because it's a lot easier to read and write the DNA that is necessary for the experiment. So both for the oligonucleotide synthesis part that pooled screens are based on, and for the Illumina sequencing part that is necessary to read out a library at the end. So if you want to do combinatorial Cas9 work, you need 2 promoters to drive the expression of 2 guide RNAs. And the constant sequence known with Cas9, which is known as the tracer RNA, is 76 nucleotides long. Whereas with Cas12a, if you want to express multiple guide RNAs, you just need 1 promoter, and you can just keep stacking the constant sequence, which is known as a direct repeat, and a guide, so on and so forth. So with Cas12a, you can do this with 2. It's kind of a pain in the butt, but it's doable. But if you want to do even more than 2 guide RNAs, the molecular biology and the sequencing would be really difficult for Cas9, whereas with Cas12a, it's trivial. You just synthesize a slightly longer oligonucleotide, and we've done up to 4 guide RNAs very easily with a Cas12a-based system. Fortunately, Cas9 and Cas12 give very similar results. It's not like they, a double-strand DNA break is a double-strand DNA break, basically. So what's shown on the left is a genome-wide screen looking at viability phenotype. On the x-axis is the effect with Cas9, and the y-axis is the effect with Cas12a. And you can see these are pretty darn well correlated results, suggesting that it's not like there are regions of the genome that are only accessible to Cas9 or Cas12a or vice versa. You get more or less the same answer regardless of which Cas protein you use. And where is this important? Well, one area where this is quite important is to study paralogs. So the dependency map, or DEP map, is a long-standing project at the Broad to understand what are the genes that are important for the viability of cancer cells. And if you go to DepMap and you search for the gene ERK1, which is also known as MAPK3, and you ask, is MAP kinase signaling important in cancer cells? DepMap tells you, no, it's not. Now, that, of course, is the wrong answer. We know that MAP kinase signaling is extraordinarily important in a wide variety of cancer cells. But on the right are the data that shows you why DepMap gets this wrong. So if you knock out only MAPK1, you don't see a viability effect. If you knock out only MAPK3, you don't see a viability effect. And so the mathematical expectation would be that if you knock them both out, nothing should happen. That's what's quantitated in the orange box and whiskers plot. But what we actually see is when we introduce guide RNAs to target both MAPK1 and MAPK3 at the same time, now we see, oh, look, MAP kinase signaling is important. So, but the reason we didn't see it with the single knockout is that the activity of one MAP kinase could substitute for the activity of the other MAP kinase. This is further compounded by the idea that if you want to do combinatorial work, we are pretty good at predicting which guide RNAs will work. I'll benchmark our success rate at about 80%, that if we design a guide RNA and we hope that it works, about 80% of the time we'll be right. But in combinatorial space, you need both of them to work, right? So let's say we're trying to target 2 genes at a time. And we have an 80% success rate for targeting gene 1, an 80% success rate for targeting gene 2. Well, that means we're only going to be right, or we're only going to be successful both times 64% of the time, which means 36% of the time, 1 out of every 3 cells, we will have failed to successfully knock out both. But with Cas12a, what we can do is actually put 2 guide RNAs into the same construct, targeting gene 1. So now only 4% of the time will we fail to knock out gene 1. So 96% of the time we'll be right. And likewise, if we have 2 guide RNAs in the construct targeting gene 2, 96% of the time we'll hit gene 2. And so our overall success rate will be 92% instead of 64%. And our fail rate will be less than 10% rather than greater than a third. So this is what we call the Informer platform. This was developed with Trevor Hart at MD Anderson, where this library, which is also available on AdGene, we target every single gene in the genome, but we also target over 4,000 paralog pairs. So we won't miss things like MAPK1 and MAPK3. And there's a lot of paralogs in the human genome. So with this sort of library design, we're much more protected against the false negatives that would arise with using by targeting only one gene at a time. Now, of course, combinatorics is important for many cell-fate decisions. The Yamanaka factors that are used to convert fibroblasts into stem cells being the most critical one. But of course, the ability to start with a stem cell and push it into all sorts of different lineages is a very important problem in cell engineering and just in understanding development writ large. So we've also recently worked to make Cas12a work not only for combinatorial knockout, but also for combinatorial CRISPR activation or turning genes on. This was recently published, and a schematic of the approach is shown on the left. And on the right is an example of it working in practice. So on the right, we're targeting 4 different markers that are easily assessed on the cell surface. So we're doing flow cytometry. And you can see that we can successfully activate 4 genes, CD4, CD97, CD274, and CD26, all at the same time. So that enables us to do combinatorial screens, not just knocking genes out, but also turning genes on. Okay, so I want to close by just pointing out that functional genomics did not start with CRISPR technology. This first quote is from Nature Reviews, that the capability to quickly and efficiently alter any gene sequence will have profound impacts on biological research and yield potential therapeutic strategies for genetic diseases. That is very true, but that was not written about CRISPR technology. That was written about TALEN technology in the year 2013. Fortune magazine noted that within a few years, this technology should yield a rough idea of what each of our genes does. Well, that was written in 2003 about RNAi, and I would note that we still don't know what the vast majority of our genes are doing. And then finally, the Scientist magazine commented on a battle of biblical proportions over the patents for a crucial enzyme in molecular biology. That was also not about CRISPR technology. That was about Taq polymerase, the polymerase, obviously, that we all use every day to do PCR. So it's just a reminder that history may not rhyme, but it certainly echoes. So, when in doubt, screen it out. There's a lot of perturbational tools in the toolbox now, and you really do learn new things. You learn new things each time you screen with a different technology. It's not the same to knock a gene out as it is to overexpress it, as it is to base edit it, as it is to look at it with combinatorics. And really, the hardest part of doing any of this is not doing the screen. The hardest part is building a model that successfully and reasonably models the disease or the process that you really care about. And once you've developed that cell model, mouse model, whatever it may be, you can do your first screen, and that's great. But then once you validate that this model is good for screening, there's a lot more that you can learn from it. So time spent developing the model is probably the most important part of getting any screen to work out well. And then you can go to town continuing to learn new things about it. So I want to thank all my colleagues in the genetic perturbation platform here at the Broad Institute. It's a really wonderful group of folks who I think internalizes this quote. Harry Truman's not our most quotable president, but I really do like this one. It's amazing what you can accomplish when you do not care who gets the credit. So we hope to provide lots of tools and technologies that are available to the world, and to see people use them and make all sorts of great biological discoveries with them. So with that, thank you so much for hanging out with me this afternoon, and I am more than happy to take any questions. Thank you. Chandra, you are muted, but I'd love to hear from you.
Can you hear me now?
Yes, yes.
Okay. Like Verizon commercial. Okay, because that's not new either. Anyways, okay. Question for you: if you were going to do an enhancer screen, meaning that you're looking for lots of enhancers across the genome, and you're trying to figure out which enhancers affect transcription more globally. Let's just say your problem as a screener is to figure out how do we target and silence each of the enhancers. Would you do that with CRISPR-I and not just one enhancer? So like can't do base editing tiling screen, but how would you do that? Would you do CRISPR-I, and where would you target, and how many would you target?
Yeah, so how to study non-coding elements with CRISPR technology is, I think, still something that is very much being worked out. And I think the challenge is none of the approaches that one could take really do what we want to do exactly. And so one would want to be very careful about, well, what are both the false positive and false negative modes of each approach? So let me walk through them and feel free to add more to my list of options. So one approach, if you have a non-coding element, one approach is to say, well, let me just cut the element out. So let me put a double-strand DNA break here and a double-strand DNA break here, and hope the whole thing goes away. So that can work. I think one challenge there is the efficiency, because one thing that will happen is if you introduce a double-strand DNA break here and one here, sometimes you'll get the ends to glue back together, but sometimes you'll get non-homologous end joining here, non-homologous end joining here, and you'll now just have like little scars flanking your element, and you won't really learn much of anything. And so it works when it works, but I think it's hard to predict when it will work well. And I think that more work needs to be done to make that technology really efficient. Another approach that you mentioned, Chandra, was, well, let me do CRISPR-I. Let me land a Cas protein on top of my enhancer and have that Cas protein fused to a crab domain, for example. People do that. I don't know what that really is modeling from the standpoint of, I mean, if what your enhancer is doing is recruiting a crab domain, then that seems like a reasonable model of what the enhancer is doing. But I think lots of enhancers don't do that. They are doing something different. And thus, it's not clear to me the relevance of landing a crab protein at a certain site in the genome to be able to say, okay, this is what this enhancer does. You would almost want to try, like, well, let me do a screen where I land crab proteins at the enhancer, but now let me land a different chromatin modifying enzyme or a transcriptional activator or whatever the case may be. And if you're just using it to understand, like, well, what does this enhancer do? It's hard to know which one would be the right answer. And so I think you can think about like the long list of things to try, and ultimately it gets, I think you would need to try several of them and then sort of coordinate and triangulate on like, well, at least this enhancer in this cell type seems to give me a reasonably consistent answer regardless of the way I perturb it. But it's a really hard question of just interpreting non-coding DNA for sure.
I'll ask more questions, but I'm going to give a second. I have another one. I'll give everybody else a chance.
I guess I have a very naive question about sort of the GPP. It looks like you have a lot of resources and can help with sort of the reagents, but do you guys do anything in terms of like troubleshooting or helping people in the development of the actual screens? Because that's a burden of activation for some folks.
Yeah, no, absolutely. And, what we've tried to do is maximize by minimizing the amount of time people need to worry about the logistics of a screen. Hopefully that expands the amount of time people can worry about thinking about, is this screen a good idea in the first place? So maybe put a different way. I think one of the biggest challenges I see with beginning a screening campaign is people are excited to do a screen. Like, oh, I'm going to do a screen. I'm going to learn all these things. Go, 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 go. And once you get going with doing the assay dev and then conducting the screen, once you're in logistics mode, it's very hard to step back out and say, like, wait, why am I doing this in the first place? And the question I often pose to folks is, okay, let's say you were to do a screen. I promise you, I will send you a hit list where something will be ranked #1. There's always something ranked #1 by definition. What are you going to do with it? And if the answer is, oh, I don't really know, or tell me the name of the gene and then I'll tell you what I'm going to do, I would say you're probably not ready to do your screen yet. I think thinking through, why am I doing this and what is the follow plan? In other words, if the screen is figure 1, what is figure 2 going to be about? And having a good sense of you don't need to know exactly, but having thought through the project more fully, I think is really critical. So those are the conversations that one can have long before you pick up a pipetter. And those are the conversations that are fun to have and fun to brainstorm with, not just people that have done screens, but people that maybe have never done a screen. And so I'd really encourage folks who are thinking of doing a screen to talk to others and really think about, okay, fine. Assume figure 1 happens and there will be resources and help and whatever, you can make it go. But what's figure 2? How are you going to turn this into a story?
Oh, also, that's a great answer. I just thought of something. I know how you have your hand up, but Chandra, maybe you want to introduce yourself because there could be people here but actually don't know who you are.
I can't fathom that, but yeah.
I mean, right? So I'm Chandra Miller. Sadie's actually a graduate student in my lab. I also direct the Center for Advanced Genome Engineering. So locally, you have some expertise here. And so if you're thinking about doing a screen, we would probably use one of John's beautiful, if it's whole genome, we probably would use one of his libraries, but we can also help make custom screens, and then we can help support. We have a very similar model of GPP. Like, we don't do the screen. We help on that end. So reach out if you have questions.
Thank you. Oh yeah, sorry, go ahead, Hao.
Oh, you're muted, my friend. Hao, you are muted.
Sorry about that. So I was saying we don't actually do screening in the lab, but we work with in vivo models. We work on the brain. So we've been using some Cas9s, but the Cas12a sounds very interesting. Do you know if there's any disadvantage using Cas12a versus Cas9 for in vivo screening, for in vivo work?
Yeah, so 2 weeks ago, I would have said that one of the main downsides to trying to use Cas12a is that there are no Cas12a transgenic mice. But in the last 2 weeks, I've seen at least 2, maybe 3 preprints describing the development of Cas12a mice. So whether using Cas9 or Cas12, if you don't need to deliver it to the cells because the mouse already has it, then that really reduces the activation engine of the experiment. So now that there are Cas12a mice coming online, I think that really puts them on much more even footing. That said, Cas9 has been used by more labs. There's more infrastructure built around it. So I wouldn't see any need to switch to Cas12a, like just for the sake of picking up a shiny new object. I think where Cas12a really, really shines is if you ever think you want to study combinatorial phenotypes, the ability to go from single gene phenotypes to combo phenotypes with Cas12a is a lot easier than it is with Cas9. But at the end of the day, they're like, if you've ever worked with RNAi, working with any CRISPR-based technology is just such a game changer that I would not argue strongly for one versus the other. It's more a question of practicalities.
Thanks. So can I have a follow-up question?
Sure.
Okay. So you mentioned delivering, and that's one of the major issues because we are actually working with rats rather than mouse. And delivering is an issue, and there are some small Cas enzymes that can be packaged into like an AAV instead of a lenti. Have you worked with those?
Yes, definitely. So just to step back and put some context on the question, AAV is a great way to deliver to a variety of cell types. And neuroscientists really love delivering AAV, but it has a very strict packaging limit. And if your mouse or rat model hasn't been engineered to express a Cas protein, and you need to deliver the Cas protein, like Pyogenes Cas9 basically fills up the entire packaging capacity of AAV on its own. You don't have room for promoters and guides and all the other fun stuff that you would want. But there's lots of Cas proteins in nature, some of which just happen to be smaller than Pyogenes Cas9. And folks have been working with those specifically for the purpose of AAV delivery. I'd say right now, the most widely used Cas protein for AAV use is Cas9 from Staph aureus. It's several hundred bases smaller than Pyogenes Cas9 and fits into an AAV. Actually, we very recently, in collaboration with Eric Sontheimer at UMass Medical and here in Worcester, worked with him on some AAV vectors to deliver Staph aureus Cas9, and that one works pretty efficiently. That said, there's hundreds of thousands of Cas proteins found in nature, some of which are even smaller. And there's reports of them working in bacteria, kinda, and then maybe if you express high enough levels of them in a 293 T cell, you can get some DNA cutting. I think why a Cas protein works in nature but doesn't port well to human cells is still largely not understood. There's some tricks that people are trying that might be helpful. So it's definitely an area where I'm giving you a long answer. The short version is use Staph aureus Cas9, but you never know when something better is going to come along. So at least keep your ear to the ground.
Great, thank you so much.
Chandra, would you agree with that?
100%.
Oh,
yeah,
good.
I mean, I think there's like these Geo Cas9s. It's like they kind of maybe a little bit work, but I think to me it's just amazing that we were so lucky that SP Cas9. Because what if it would have been Geo Cas9 that they did first? We wouldn't be where we are. So it's serendipity, I guess. But
very much so. Very much so.
Okay, I'm gonna ask one last question. What are your thoughts? So like I've seen some prime editing screens. Seems super challenging. Prime editing's getting a little better. What are you guys doing? What are you thinking about GPP wise? So what prime editing screens that I've seen is basically they like optimize and optimize and optimize and optimize to figure out which peg is going to work for like 150 base pair region that they're going to tile. Do we care to do that, or is it better to just do a base editor screen? I mean, now functionally base editor screen, but what are your thoughts, I guess?
Yeah, so prime editing is a technology developed by David Liu's group a couple of years ago. And unlike base editing, that uses a deaminase domain hovering over the single-stranded DNA, and thus you're limited to the changes you can make to what that deaminase can do. With prime editing, you're using reverse transcriptase to ultimately introduce your DNA of interest into a precise spot. So you can make basically any small nucleotide change, insertions, deletions, point mutations, whatever the case may be. So it is more broadly, you can kind of do anything you want with it. But as Chandra's question very much got at, the efficiency of prime editing still seems to be pretty darn low. And for screening purposes, we're taking what I'd call a watchful waiting approach, where we keep looking to see, okay, if this is new technology that some lab develops to improve the efficiency, is it good enough for a screen? Now, it might be completely fine for engineering a cell model. Like you want to introduce a flag tag into the C-terminus of a protein of interest. Prime editing might very well be the way to go in a particular cell type. But to screen across lots of cells, you need the per cell penetrance of the guide RNA that you introduce to be quite high. If it goes into 100 cells but only introduces the edit into 5, you don't really have any signal for your screen. So where I think it has demonstrated some utility, and I very much appreciate your point that it's a lot of work to get there, is if you have a very strong positive selection phenotype, where even if you introduce the change in only 5% of cells, if those are the only cells that survive a very strong selective pressure, then you'll be able to identify them because everything else will be dead. But what I don't think prime editing is really capable of doing yet is doing anything in the negative selection direction, which means find the changes that lead to like loss of function or depletion phenotypes. And that's where I think most of the interesting phenotypes are, not all of them, but most of them. So, we're continuing to watch and see what goes. But if I had a protein of interest today and I wanted to tile across it, I think you're just going to get much better activity. I'd rather have the higher activity of a base editor, even though it's lower resolution, than lower activity and higher resolution with a prime editor. But that might change next week.
And on those base editing screens, do you ever, how often would you do, like, I'll do a base editing screen with an A base editor and then I'll follow it up with the same gene with a C base editor? How often does that happen? Or do you feel like you get enough with one or the other?
We're generally doing both just to, provide more confidence and get a little more resolution. I think if you were interested in doing lots and lots and lots of genes, you could, if you had the budget and bandwidth or whatever to do 20 genes with one base editor or 20 genes with 2 base editors or 40 genes with one base editor. If you really cared about genes 21 through 40, it might be better to do them with only one base editor, but in other circumstances, it would be better to just do both. But that's really, more a question of what biology you're asking. I see a question from Eric in the chat. Is prime editing being done with Cas12a? I'm not aware of any particular reports of prime editing with Cas12a. I might be wrong about that, but the Cas12a, the way it cuts DNA is fundamentally different from the way Cas9 cuts DNA. Cas9 opens up single-stranded DNA, and the active site is sort of in the middle of where the guide binds, and that's pretty fundamental for both the base editing and the prime editing approach. With Cas12a, the active site is sort of at the end of the guide, and you don't get a nice single-strand DNA bubble or a single-strand DNA exposed nearly as much as you do with Cas9. So both base editing and prime editing base editing has definitely been less efficient with Cas12a, and I haven't seen any prime editing done with Cas12a. Don't get me wrong, I would love to do like multiplexed base editing with Cas12a if I could, and just see how SNPs interact with each other, and that would be really fun. But just the efficiency is not there right now.
Okay, thanks. Yeah, I was just curious if it's technically feasible, because the multiplex aspect would seem like that might be something you could help to do, like twin prime editing or some other things like this to make them more efficient.
Yeah, exactly.
Any more questions? Well, I guess you get off easy. Yeah, thank you. This is amazing talk. It's really great to see the progress of all these technologies, and yeah, I'm sure there's a lot of people here that are sort of pondering or considering how they can fit these types of screening into their research. But so hopefully more people will reach out to you and also to Chandra. And see, because it really is amazing what you can do. And this could be a good companion, I think, to people who are also interested in like screening genetic populations. That's an obvious one. And then understanding signaling, how genes work. So really awesome. Thank you so much for taking the time out to meet with the grad students and us.
Absolutely. Wonderful to see you all. Thanks again, Sadie, for the invite. Wonderful to see you.
Of course. Thank you.
Thanks for hosting, Megan, and I'll see you all around. Have a good one. Bye-bye.